So how does the process of LTP work? Well, the first thing to note is that there really isn't just one kind of LTP. There are many different mechanisms that can lead to the potentiation of synapses, and studying them is still very much an active area of research. Broadly speaking, all LTP results in stable changes in a host of molecular and structural features in the synapse, including the axonal bouton, the dendritic spine, and in particular, the proteins of the postsynaptic density. To make a synapse stronger, we either have to make more neurotransmitter be released, increase the number of receptors on the postsynaptic side, or somehow make the postsynaptic receptors produce a larger effect. All three of these mechanisms have been observed in nature, as have combinations of these mechanisms. Another important axis of variation in LTP is how the potentiation is triggered or induced. LTP can be broadly classified into two categories: Hebbian or non-Hebbian LTP. Hebbian LTP follows Hebb's cells that fire together, wire together rubric. Hebbian LTP is the easiest to think about, and it's the classic form of LTP that you'll find described most often in neuroscience textbooks. We'll spend some time describing a particular kind of Hebbian LTP called NMDA receptor-dependent LTP in a moment. But before we do, it's worth noting that non-Hebbian forms of LTP, including explicitly anti-Hebbian forms of LTP, are prevalent in the nervous system as well. Anti-Hebbian LTP, as you might guess, works in the opposite direction from Hebbian LTP. When the presynaptic and postsynaptic neurons fire together, the synapse tends to weaken. The diversity of different kinds of LTP no doubt play an important role in supporting the diversity of different types of learning and computation performed by the brain. As we alluded to earlier, one of the best understood forms of LTP is NMDA receptor-dependent LTP. Since it's in many ways the canonical example of Hebbian LTP, we'll focus on how it works for the rest of this lesson. In Hebbian LTP, a synapse is strengthened when the pre- and postsynaptic partners in the synapse fire in close temporal proximity. What fires together, wires together. How does nature implement such a rule? One answer is the NMDA receptor. The NMDA receptor is a special kind of glutamate receptor. It's named for N-methyl D-aspartate, NMDA, a substance that binds to the NMDA receptor, but not to other kinds of glutamate receptors. NMDA itself isn't a naturally occurring substance, and NMDA receptors didn't evolve to bind to it. Rather, we call the NMDA receptors NMDA receptors simply because scientists use this synthetic substance NMDA to identify the receptor. This is a somewhat confusing naming convention, but it's a common one, and we're stuck with it. If you recall our previous unit on excitation and inhibition, the AMPA receptor got its name by a similar route. NMDA receptors are complicated molecular machines, and the structure and function of the NMDA receptor answers a first key question about how Hebbian plasticity can be implemented. How does the cell know when the pre- and postsynaptic neurons fired in close temporal succession? The prominent neuroscientist Eric Kandel did seminal work to unravel the beautiful molecular choreography of these mechanisms, earning him a Nobel Prize in the year 2000. Let's start with the basics of the NMDA receptor. The NMDA receptor is an ionotropic receptor. That means when it's open, it lets cations, and most importantly calcium, pass through it. But here's the thing, it's actually a bit tricky to get it to open all the way. For one thing, the NMDA receptor requires not one, but two ligands to open it. Both glutamate and glycine must bind to the channel to open it. However, even more trickily, the NMDA receptor is both ligand-gated and voltage-gated. This is the key to how NMDA receptors can enable Hebbian-style plasticity. When the neuron is at rest, the NMDA receptor channel accepts a magnesium ion that blocks the channel's pore. However, when the neuron is depolarized, the magnesium blockade is repelled and ions can flow through. Thus, to open the channel, we must both depolarize the membrane and activate the channel with neurotransmitters. This provides the requisite fires together, wires together property that we've been looking for. We need to depolarize the postsynaptic neuron to release the magnesium blockade, and we must activate the presynaptic neuron to release neurotransmitter to provide the ligand. Now you might be wondering how we would depolarize the postsynaptic neuron in the first place if NMDA receptors are so finicky to open. The answer is that we rely on another kind of ionotropic glutamate receptor, the AMPA receptor, to depolarize the membrane. You may remember from a previous lesson on excitation and inhibition that AMPA receptors are common excitatory glutamate receptors in the nervous system. AMPA receptors are cation selective channels, and when they're made with the appropriate variant subunits, they let sodium and potassium through, but block calcium. Thus, AMPA receptors, both at the synapse of interest and at other synapses, depolarize the cell but don't let calcium through. And NMDA receptors carefully allow calcium to pass through when both the pre- and postsynaptic sides of a given synapse are active. So why is letting calcium through so important? Well, calcium, it turns out, is a very important ion for signaling within neurons, mainly through the action of an enzyme called CAMK2, which stands for calcium calmodulin-dependent protein kinase 2. This influx of calcium activates two downstream biochemical and metabolic mechanisms. First, it triggers molecular cascades that phosphorylate, that is, add phosphate groups to AMPA receptors, which in turn makes them open more easily. This process is known as AMPA receptor phosphorylation and allows the receptor to be more sensitive than normal after LTP has taken place. In addition, calcium influx triggers another cellular mechanism that switches on further production and speeding up of the trafficking of new AMPA receptors, which leads to the insertion of new AMPA receptors into the postsynaptic membrane of the synapse. The mechanisms by which this trafficking takes place are still a subject of significant research and debate, but it's worth taking a step back to consider what a remarkable feat potentiating a signal synapse represents. Molecular signaling cascades can easily upregulate or downregulate the expression of various genes, but such regulation necessarily applies to the whole cell at once. We can tell the cell to make more AMPA receptors, but targeting those receptors to a specific synapse is another story altogether. NMDA receptor-dependent LTP exhibits several very interesting properties, including input specificity, associativity, cooperativity, and persistence. Let's briefly walk through all of these individually to give you a flavor of the complexity of synaptic potentiation. First, once induced, LTP at one synapse largely doesn't spread to other synapses. Its input is specific. This is critical since neurons can have thousands of synapses, and when we potentiate a particular synapse, that one synapse operates largely independently of the other. The effect doesn't leak. A second interesting property of NMDA-dependent LTP is associativity. When weak stimulation of a single input is insufficient for the induction of LTP, simultaneous strong stimulation of another input may induce LTP at both inputs. This follows from the voltage-gated nature of NMDA receptors. A strong input elsewhere can depolarize the cell, allowing NMDA channels at sites with weak activation to open. Likewise, LTP can be induced cooperatively via weak stimulation of many synapses. In this case, collective action of AMPA receptors at many synapses depolarize the cell, enabling NMDA receptors at many sites to be activated. The final key property of LTP is persistence. It can last for minutes, to hours, to months, or even longer, and it's this persistence that separates LTP from other forms of synaptic plasticity.